Hello everyone and welcome to another episode of Nowhere Yudarris. Today we'll be speaking about proteins in the biochemistry kind of theme or subject. We're going to speak about 5 types of protein and what each type consists of and what contributes to, etc. etc. So stay tuned, don't forget to like and share and subscribe as well, and check out our small TikTok reels, and let's get started. So there are 5 types of non-enzymatic proteins. There are structural proteins, motor proteins, immunoglobulin, binding proteins, and biosignaling. Each one of them is unique and have a specific function. So let's start with the structural protein. These types of structural proteins are what make up a structure of a lot of tissues and cells and act as supportive backbone. So we're going to start with collagen. So it makes up extracellular connective tissue and provides strength and flexibility. You know how collagen is sometimes in between some of your tissues, and it's part of your connective tissues. It's one of the main important factors in the strength and flexibility. Next, you have elastin, which makes up extracellular connective tissue for stretch and recoil functions in the tissue. I want you to imagine elastin. It sounds like elastic, so that's how you memorize it. Kind of stretches and recoils. Then you have keratin, which is an intermediate filament protein in epithelial cell, and it's a primary protein that makes up your hair and your nails. I want you to look at your nails, and that's keratin right there. Next, you have acetin, which makes up microfilaments and thin filaments in myofibrils, and its polarity allows motor protein to travel along acetin filaments. So acetin makes up microfilaments and thin filaments in myofibrils, and it kind of co-works with the motor proteins to allow them to travel along the acetin filament. Next, you have tubulin, which builds microtubules, which are important for structure support and chromosome separation in meiosis and mitosis. And an important part of meiosis and mitosis is kinesin and dynein as well. Tubulin kind of also co-works with motor protein, kinesin and dynein, which we'll be speaking about shortly. Next, we have motor proteins, which are responsible for muscle contraction and cellular movement, like you see in a cilia or a sperm. So you see how they have their tails, for instance. That's motor proteins helping them move. So they act as ATPase is the power up the motor function. So motor proteins contribute to the motor function of um, I want to say cells or organisms such as cilia or sperms. And important parts of motor protein, or important types, sorry, is myosin, which interacts with acetin for cellular respiration. So remember, we said acetin co-works with motor protein, allows it to go on its acetin filament. So that happens using myosin. And we spoke about kinesin and dynein. They're associated with microtubules and with meiosis and mitosis. And then you have cadrins, which are glycoproteins that hold calcium-dependent cells in adhesion. They are responsible for cell-to-cell adhesion in animal cells. And you have selectins, which bind to carbohydrate molecules that project from cell surface, and it's found on white blood cells. So selectins are also part of the immune system, which we have an episode on that. Um, Next, we have immunoglobulins, which are also part of the immune system. They're part of the immunoglobulin protein system. By the way, selectins are part of specific immune system. So immunoglobulin, humoral immunity, are protein produced by B cells that neutralize the targets in the body. They have the antigen binding region that allows them to neutralize the antigen and mark the pathogens to be destroyed by white blood cells. So that's an immunoglobulin structure, a basic one. So produced by B cells to neutralize the target. They have the antigen binding regions here, and agglutinate antigen and antibodies into a large protein complex to be phagocytized by macrophages. So agglutinate is basically they kind of a collect like the antibodies, and they can collect the pathogens into one large complex so they can be eaten by macrophages. In simple terms, it's not really eaten, but you know what I mean. Next, we have the protein biosignaling. They are cells that receive and act on a signal, such as ion, enzyme-linked receptors, and G protein. So ion channels are facilitated diffusion and passive transport that allows membrane proteins to serve as channels for substrate to avoid hydrophobic fatty acids. So a lot of cells, a lot of a what do you call it? A lot of substrates, they can diffuse across the hydrophobic fatty acids. So that's what the ion channels are there for, to help them diffuse. So you have three types: ungated, voltage gated, ligand gated. So the ungated is basically no regulations. You know, it will let in most cells, and it's associated with potassium channels, thus increasing. There's a lot of potassium. Why is that associated with potassium? Because potassium is one of the largest atoms. So if it allows such a large atom, it will probably allow other things as well. Next, you have voltage gated, and this is really related with the action potential regulation, such as the sodium potassium channels found in the sino-arterial node heart, or associated with neurons. We also have an episode on um, action potential regulations. Next, you have the ligand gated channels, and these are the binding of a specific ligand that opens or closes the channels as neuромembrane junction, postsynaptic membrane, for instance. So these are ligands that bind to the channel, and opens or closes the channel depending on what kind of pathway there is. So then you have next the enzyme linked receptor, and they display catalytic activity in response to a ligand. So ligand gated channels are kind of related to enzyme linked receptors. And you have membrane spanning, which anchors receptors in cell membrane. And you have the ligand binding, kind of related to the ligand gated ideas, that stimulated by an appropriate ligand and induces a conformational change that activates catalytic activities, for instance RDK. So basically a specific ligand binds to the enzyme and induces a change in its active site and its shape, so it can activate catalytic activities, or so it can kinda accept a specific type of substrate. And then lastly, you have G protein coupled receptors. These are transmembrane receptors that use GTP, responsible for the activation or inhibition of secondary messengers. So remember with me, G proteins use GTP for secondary messengers. So a GS stimulates adenylate cyclase that increases CAM in cell, and GI stimulates adenylate cyclase to decrease CAM in cells. GQ activates phospholipase C, which cleaves phospholipids from membranes to for instance, create PIP2 to DAG to IP3, which opens calcium channels and increases it. So GQ kind of is responsible for the increase of calcium channels because it activates a certain phospholipase, which cleaves phospholipids, and then it results at the end in the opening of calcium channels. So GS increases CAM, GI decreases CAM, and GQ increases the calcium channels, or calcium technically. SDS page, which denatures protein and masks native change to show size comparison. Functional protein cannot be recaptured. And lastly, we have electrophoresis method, which is used to separate proteins using a polyacrylamide gel. large molecules move slowly, and smaller molecules to medium molecules, you're gonna see them move faster down the gel. I'm gonna make an episode on lab techniques soon, and hopefully you enjoyed this video and you found it informative. Please leave a like and leave any comments, and thank you so much.